Good morning, Suzuki Science, and I hope that you guys had a pleasant weekend. This is number 2 video. The first video was on the review from what we did before the break. We had a whole story, the cell story. I always like to start my lessons going back to the beginning and moving forward. If we went back to the beginning of cells, we know that the cell is the smallest unit of life. We know that all cells come from pre-existing cells. We know that the cell is going to be so small that we need some special tool to look at it. So we played with the microscope before the break as well. Sadly, you will not be playing with the microscope this year. You'll have to wait until next year. If you decide to take grade 11 bio, then you get to play with the microscopes again. We used the microscope to see the cell because it was too tiny. There's a limit on how big it can grow. We had a whole lesson and a lab, which most of you, well, many of you did submit, some of you did not. The BEAT lab allowed you to explore why certain cells can only have a certain size. If they get too big, it's incredibly inefficient. And so when a cell gets too big, it will either die or more likely it will replicate. That process is known as mitosis. The cell cycle defined by G1, S, G2, and mitosis. At the end of it, the cell will have 2. And at the end of that, there'll be 4 and so on. So mitosis is a prequel to what we are doing now. Sometimes when a cell divides, however, there is a problem. And when a cell divides and there's a problem, you might end up with what you see on the screen here in front of you, cancer. Now, in terms of this lesson, my suggestion with this lesson is you're going to see several things. You're listening to the lesson, clearly, and there's another document, which is your document that's the Google Doc, and this is where you're going to get most of your notes. This first page is just text, and it is your note. So you can use this in lieu of your textbook. We are ending our kind of cell division story with cancer. What happens if things go wrong? So as you look at this, all this stuff here is all things that you should know. There's a nice little graphic. It's super simple. It shows what a cancerous cell and normal cells look like. And then it ends off with a couple more paragraphs. You'll see that this lesson is divided into 4 parts. So when you go through this, we want to start with part 1. In part 1, I'm asking you to come up with a definition of cancer. So I'm going to help you learn cancer, but I want you to derive the definition first. So everyone is doing part 1. To do part 1, you click on this link. When you click on this link, so you access this document again from the Google Classroom, and when you click on that link, you're going to get to a screen with a series of videos. And those videos are right here. So what you're going to do is you're going to watch 5 videos. Each of them are 30 seconds to 1 minute in length. And on your document, you'll see that there is a sentence, and the sentence is missing a word. So in animation 1, I'd like you to watch the video and listen, and you're going to complete the sentence. Cancer involves there's an adjective, division of body cells. And you can do that for animations 1, 2, 3, and 4. When you get to number 5, please notice that there's no sentence here for you. It's just a statement. So you're going to basically write down the definition of what cancer is. And you're going to get that information from your summarization of 1 through 4 and particularly 5. Don't worry if you're not able to come up with a great definition. That's perfectly fine because I have another sheet that will have the answers for you. You'll see it on the Google Doc. Okay. Once you've done those 5, to end off part 1, I would like you to click on that second link, and what it will do is it will open up these news alerts. And so these news alerts propose 4 different ways that you could get cancer. So you just click on the link. It's a YouTube video.
I have been studying the various forms of cancer.
Yeah, we don't need to listen to it together. You'll listen to it on your own. And after you listen to like that 46 seconds, then you're going to write down that possible cause. So you'll have 4 possible causes. So at the end of this part 1, you'll have a definition for cancer. You'll know why cancer occurs, and you'll know 4 things that cause cancer. So that's a great start. These lessons that you're doing each week are typically layered. So there's a minimum that you can do, and that typically will be about 3 hours, and then we'll, maybe a little less, honestly, and then we'll layer other work on top. And depending on how much you want to know, how engaged you are in the learning, you can decide how much you want to do. There will always be some kind of product or something at the end of the week, like I mentioned last week, and I'll get to that eventually. So in this particular lesson, there are 4 parts. That was part 1, so part 1 is done. Part 2 is working your way through the presentation. The presentation has 2 forms. There's a long form and the short form. The long form is about half an hour longer than the short form. So I have this whole preamble. I talk a little bit about the history of cancer. And you do not have to listen to that. It's just for interest and engagement. So you can totally skip that. I'm going to record the short form in a different video. So I will let you know when you can stop listening and fast forward on to the next one. Or if you're engaged in the grade 10, you want to be better prepared for grade 11 or grade 12. If you want to be, actually have something to do for this week that's interesting and engaging, and elicit some conversation from your parents and your friends about this concept, then I encourage you to listen to the long form. But really, this is just an excuse for me to talk. Now, once you finish the video, the long or the short, there are 2 activities. One of the activities, once again, is to engage you in the learning process. If you complete part 3, then you will be better prepared for the final evaluation. You'll be much better prepared for grade 11 and grade 12 and beyond, and it's just interesting. But it is optional. For those of you who think that grade 10 science might be it for you and you just want to move on to something else and you have better things to do, like sit in your bed and watch the wall, then you might want to, you could skip part 3 and just move right to part 4. Everyone's doing part 4, and I'll talk about part 4 a little bit later. Okay, so now I'm going to move back to the presentation, and the presentation is going to be the long form. So for those of you who do not want to listen to the long form, or at any point you're done listening to the long form, you can stop and you can open up the next presentation. Okay, a copy of this presentation, the long and short combined, are also in the Google Classroom. Okay, long form, cancer. So you've had some time to look at the cancer slide that I've had here and look at some of these quotes. Cancer's been with us since, the dawn of human history writing. Our records show that there were people with cancers, and they were not called cancer, but it was clearly the same idea of what we understand cancer is today. So I look at this story. Look at this as a story. Medicine begins with storytelling. Patients tell stories to describe illness. Doctors tell stories to understand it. Science tells its own story to explain diseases. This story of one cancer's genesis, beginning of carcinogens causing mutations in internal genes, unleashing cascading pathways in cells that then cycle through mutation, selection, and survival, represents the most cogent outline we have of cancer's birth. That's a sophisticated paragraph. This is actually a paragraph taken from a book called The Emperor of All Maladies. This is an excellent book. If you're ever interested in knowing about cancer, listen, a biography of cancer. Look at the ratings. Look at the number of people who rated it. This is the Canadian site. So if we went to the American site, it's probably even more. Okay, no, it's not. But either way, it's an excellent book. If you want to know more about cancer, then I would encourage you to go out into the library and borrow it. So when we look at the beginning of history, look at that. 2,500 years ago, we have this original text, Egyptian text, and it describes, I can't read this, but somebody says that this paper is talking about a bulging tumor in the breast, which is a clear indication of a cancer. Breast cancer was and is and continues to be a very common form of cancer, like touching a ball of wrappings. I'm going to fast forward about 4,000 years, maybe a few more than that, and we're going to get to the 1940s, the age of the antibiotics. So we had a lot of diseases, and once upon a time, it was the infection that would kill you more often than something else. In the world wars, more people died from infections than they did from the bullets and the blunt force trauma. But then penicillin came along, and penicillin, it was able to fight a lot of bacterial diseases. That was in the 1940s. And in 1943 was streptomycin, another antibiotic that would fight another whole host of bacterial diseases. And then tetracycline in 1948. Typhoid, you might have heard of Typhoid Mary and her story. Was a disease that was very contagious and it killed many people, millions of people. But In 1942, with the antibiotics, its incidence was greatly reduced. TB, we still have to this day, like typhoid as well, actually. And in the 1910 to 1940, the number of people that were killed by the world's most infectious killer was reduced in half, and still to this day continues to be less. Polio was common up until the 1950s, and then we had a vaccine that was created. So that era from 1940 to 1950, the age of the antibiotics, there were so many developed. Sadly, in the past 15 or 20 years, we've had very few antibiotics developed. And as you look at this story, why am I talking about it? Well, there's something missing in this story, and that's cancer. Cancer was not cured at this time. We still don't have a cure for cancer. Cancer has been very difficult to deal with. Local cancers, yeah, we can treat those, but how do we treat local cancers? By taking a knife and cutting it out. Or, even now in modern days, taking a radiation and radiating the cells and killing them with radiation, and sometimes killing the cells around it. So these are two very blunt, big, giant ways of removing the cancer. There's something missing from this story, though. For anyone who knows anything about cancer, you also might know that we use chemotherapy. So chemotherapy is a very interesting story. Lymphomas. A lymphoma is a cancer of the blood. And I have a little page here with a little interactive. And what happens is your white blood cell count is incredibly high. You produce way more white blood cells than you're supposed to have. And there's so many of them that they actually interfere with the other blood cells, and they don't work properly anyway. So these lymphomas are a common cancer, and they're very problematic, as you can imagine. And it was found that folic acid, yes, the same folic acid that we tell pregnant moms to have to help with the development of the fetus, the folic acid actually increases the incidence of, or the development of, these cells that produce these lymphomas. And so as scientists, and I'm going to say scientists because there's a group of scientists that contributed to this knowledge, as scientists, we're trying to figure out, okay, how do we stop these cancers? Okay, folic acid, that seems to increase it. Then what can we do to stop it? And they found this thing called Marmite. It's this yeast extraction. It sounds very pleasant, I'm sure, to many people. But I guess I'm not the biggest fan. But this Marmite, it did something. It somehow prevented the folic acid from being incorporated into these cells. And from this, this German Nobel-winning scientist named Paul Ehrlich, he discovered or thought to himself that there's got to be some anti-folic acid that he can create that would stop folic acid from doing its thing. And if he could do that, then he could stop these lymphomas in their track. And he figured there's got to be some kind of drug that could kill bacteria without killing the cells of a human. And this term, this drug, the thing he hadn't discovered yet, he called a magic bullet. And so he spent many years working on these magic bullets. Compound 606 is actually after 605, which is after 604, before 607. So it was just a generic name. And it was a drug that they found as a dye that was able to kill bacteria. But it was very toxic. What this salvarsan did is it did kill some things and make people live a little bit longer, but the side effects of the drug were actually pretty bad, and people died from the drug itself. So that wasn't great. Mustard gas is a very interesting history. Mustard gas was used in the world wars to kill people. This German scientist, Haber, who also contributed some great knowledge. He allowed us to extract nitrates and create nitrates without having to rely on bird poop and bat droppings. But he also used these nitrates to create this gas. And this gas was then used to kill people. And so the Allied forces, the very first time they got exposed to this yellow-green mist that was sitting at their feet and creeping up, that went through their clothes and through their masks and killed thousands of people. So what do you do? So World War I ends, and World War II is on the horizon. And the Allies are like, okay, we need some kind of antidote to protect our people from this mustard gas. And so what they did is they started experimenting on these people. And when they looked at them, they found out that a lot of these people from World War I who'd been exposed to the mustard gas who didn't die, their immune system, their immune cells, their white blood cells were basically non-existent. They were completely reduced. And they're able to deduce from this that the mustard gas it targeted the white blood cells. And so if the white blood cells could be killed by the mustard gas, they figured, well, the cancer cells, maybe we can kill those with mustard gas as well. And there's this person, his name was JD, he just had initials. And he had this massive lymphoma in his jaw. It was so big that he couldn't open and close his jaw. They stuck a straw in and he sucked a liquid diet. He was riddled with cancer. He had these huge cancer lymph nodes in his armpits. He couldn't move his arms. He couldn't cross his arms. He was basically encased in cancer. And so they experimented on him. He was the first one. They took mustard gas and they started injecting his cancer lymphomas with this mustard gas. And he was able to speak. He was able to eat. He was able to move his arms. The mustard gas was actually successfully targeting the lymphoma. Sadly, though, mustard gas is not very specific, and after 6 months of treatment, eventually he succumbed to his cancer and he died. But this gave rise to other ideas. We now learned, the scientific community, that a chemical could be used to kill or destroy cancer. This is the birth of chemotherapy. Mustard gas, originally designed to kill people, could be used as a chemotherapy to save people. So if we fast forward to another scientist, he took this same idea, his name is Haddow, and he modified the mustard gas, he modified the chemical, and he created this chemical. And today there's a drug called chlorambucil, which is only a few atoms different from the drug that Haddow created, and is used to treat leukemia and non-Hodgkin's lymphoma successfully. People with leukemia and NHL are able to survive more than a year, more than 5 years, more than 10 years. They can survive almost indefinitely with these new drugs, which is pretty amazing. So from the mustard gas to the modification to Haddow to what we have today, chemotherapy was born. Now, how do you know you have a cancer? So if you go to the doctor, they'll do some blood tests and they'll find certain proteins circulating in your blood. And these are tumor markers. When cancer cells are doing their thing, they tend to release these proteins into your blood. And these proteins can be tested for and you can be determined that you have a cancer. And if you have these high levels of these proteins in your blood, then they'll often tell you to go through certain, sometimes invasive tests. And so, yes, it could save you if you find out that you do have this high level and therefore there's a cancer hiding inside of you that's asymptomatic. You probably know that word now from COVID and Corona. And they're able to then take measures. But we've also found that in many cases, these protein tests don't necessarily indicate something needs to be done. If you did nothing, your body might be able to fight it. Suddenly, if you have a high PSA test that says you have prostate cancer, and then you go through the stress of thinking that, oh no, I'm going to die, and I better get my prostate gland removed, which is a invasive operation, then other things can happen. You get secondary infections and lower quality of life, when in reality, the PSA level might have been due to you exercising, or another medication, or an infection of some other sort. So you have to be really careful when you get these tumor markers and you get these tests that it's actually indicating. And if you do have a cancer of some sort, that it's actually worth your while removing it. Because in many cases, depending on what kind of cancer it is, it might not be. Here's a new word for you: cellular hyperplasia. That's another way to describe cancer. So what is the cause of cancer? It seems relevant, it seems possible and plausible that the cause of cancer is from outside your body. And as you start weighing between a bacteria causing a cancer or a virus causing a cancer, well, we already know at that point that there was a chicken that had a virus inside of it and it caused the cancer. And HPV, the human papillomavirus, we know that most viruses don't cause cancer, luckily. Viruses, to cause a cancer, have to go through these 4 steps, and most of them stop at stage 3. They will not get to stage 4. HPV, Epstein's-Barr, the hepatitis, the HIV, they can cause cancer. We know this. So because viruses can cause cancer, and we've seen it happen, it was thought that viruses might be the cause of cancer. Some other scientists, though, after the chromosomal theory was put forward, the idea that, we talked about Mendel in grade 11, where Mendel was a scientist who was able to talk about genes being transferred from one parent to the offspring, and what was those genes packaged in chromosomes. We talked about that before the break. So this chromosomal theory was put forward, and then someone suggested that, no, it wasn't viruses on the outside that caused the cancer. It was actually something inside the cell that caused the cancer. So here we have two options. Either a virus on the outside that can make you sick might cause the cancer, or something within the cell itself might cause the cancer. So when the scientists had those two ideas to think about, already knowing that polio, herpes, smallpox, and many other things were caused by viruses, it was kind of a no-brainer. It's like, okay, cancer caused by a virus. We're good. Coming back to this chicken. So there was this virus that caused a cancer in a chicken. And the retrovirus here, RSV, they took it out of the chicken, they ground it up, they kind of liquefied it, and they put another chicken, and then this other chicken, it developed a cancer as well. And so this retrovirus was then implicated in cancers. And so this particular scientist, his name's not important, he spent his life looking at retroviruses, and he was sure that retroviruses were the cause of cancers. It so happens that he died around a year or two after another major retrovirus disease came into the stage, known as HIV. But that's kind of irrelevant to what we're talking about. Through more studies and research, they determined that the virus itself caused the cells to change genetically, and it caused the cells to replicate very quickly. So the virus itself changed the DNA. That's what they discovered. I skipped a whole bunch of learning to get to that point. And these two scientists who worked on this, Varmus and Bishop, they found a human equivalent to this virus gene. And they also found the same gene in pheasants, turkeys, mice, emus. And this gene, they determined, was probably inherited from a virus, some virus way back in our history, interplayed with the host. Viruses are always exchanging DNA with their host, and it inserted this gene into our system. And when this gene gets into our system, any slight mutation of this gene causes cells to replicate like crazy. And so these scientists were able to come to the conclusion that a gene defect caused cancer. And when the gene was mutated, the cell replicated like crazy. And this was great because it meant that now we understood that viruses can cause a genetic change, along with other mutagens, including smoking and various environmental chemicals. You watched the video at the beginning with if you did the video with the chimney sweeps and the environmental chemicals that cause the cancer. So now we know that when a normal gene undergoes a mutation, that it can turn into an oncogene, and an oncogene is a cancer-causing gene. So this is huge. So this is finally us determining that cancer is a genetic disease. So at some point later on, the scientists developed this mouse. It was the world's first patented mouse. And on this mouse that had most of its immune system removed, they were able to experiment on a number of different gene modifications and produce all these cancers. Okay. I don't know if you've ever heard of 6 Degrees of Separation. So this is a movie from before you were born. There's this guy named Kevin Bacon. And there was this, it's actually a game you could play online as well. 6 Degrees of Separation from Kevin Bacon. And the idea is that Kevin Bacon knows every single major celebrity, and you can go back in 6 different steps. So one person might know another, and that person might know another, and this person might be the mom or father or banker, and eventually 6 steps away you can connect Kevin Bacon to every single celebrity. If we do a science example, we can look at Stephen Hawking, famous scientist, and we do 6 Degrees of Separation. So this is a popular meme, a popular idea that you see throughout the media. So you can do the same thing here with cancer, 6 degrees of separation for cancer. There's all these different genes, and it's thought that you can take any gene and you can go 6 steps away before you get to a proto-oncogene, some kind of gene that might cause cancer. Any one of these little steps here, if something goes wrong with them, you might end up with a cancer that causes the cell to replicate like crazy. That's pretty amazing. So knowing this concept, there's a company known as Genentech. It sounds like something from Jurassic Park. And they started looking at these genes. And so they had these big, massive databases of genes that they were exploring. And they knew that there's this one gene that caused breast cancer. And it was called the HER2. So if you had HER2, you were likely to develop, it was very possible you develop a breast cancer. And they developed this drug that was able to target this gene. And therefore, this Genentech, there's a whole story that goes along with it as well, they developed Gleevec. And Gleevec targets the HER2 gene, and it stopped the persons from getting breast cancer. It was amazing. So brand new drug. So it's no longer the dyes and these toxic chemicals, and you have to rely on the knife, with this new weapon against cancer. But cancer is inimical, another word of the day. It's sinister. It evolves. It changes. I don't know if you guys know Lewis Carroll and Alice in the Wonderland, but there's a scene where Alice is running away from the Red Queen, and the Red Queen is trying to catch Alice. And she makes a comment about how she has to run faster just to keep up and keep away from the Red Queen. And the same idea happens here, is that cancer keeps on changing. And even though we have a drug that might destroy this cancer, you just need one slight little tiny mutation to occur that will allow that particular cell to replicate through mitosis and produce many copies of itself, and then suddenly the cancer's back. So what do you do in this story? So the multiple myeloma story. So this is a classic cancer and is a great success story, where they developed several different gene-targeted cancer drugs: Velcade, Phthalimide, and Revlimid. And every single one of them targets a different gene. And so here you have this blood cancer, and you use this one gene cancer drug, and it targets the cancer, and the cancer gets squashed, and 99% of it's gone. And the people are feeling good, they're able to operate properly, but then that 1% that survived, it starts to replicate through mitosis and produce a whole bunch of other new cells. All right, so then they give them the next drug, the Velcade, and the same thing happens. They squash 99% of it. And then they give them the next drug. And they found by using 3 or 4 of these gene-targeted drugs, they did a very good job of completely eradicating the cancer forever. It was excellent, right? So that brings us to the end of this first long, and I would like to end with this word. So this is another word, phrase from that book, Emperor of Melodies. I cannot speak French, so I had to look up how to say this properly. Let me bring this over here, and I'll play it for you guys.
Fin de siècle. Fin de siècle. Fin de siècle. Fin de siècle.
All right. So what's that saying? If you translate it, it's telling us that cancer resides at the interface of society and science. Biological challenge involves harnessing the fantastic rise of our scientific knowledge, and social challenge involves forcing people to confront their customs, their rituals, and behaviors. So, smoking cigarettes, vaping, not being healthy and exercising, and obesity, eating the wrong types of foods, anything that depresses your immune system. We know that these things increase the potential for cancer. So we do need to change our behaviors. There's a time when people thought it was cool to smoke. And even though the science has irrefutably stated that this increases your risk of cancer, people insist on doing it. And if the cancer does happen, then hopefully something in science can help change that and erase the cancer from your body. This is the end of the long presentation, and I will do the short presentation in just a moment, and you can tack it on after this. Thank you, and good day.